Dear student, today we will discuss about the seed testing in seed technology course which has been offered during fourth semester of B.Sc AG degree program. Seed testing is very much important for to assess the different component of seed quality. We know very well, we discussed earlier in the seed quality, and there are different components of seed quality which can be only assessed by the seed testing. So define the seed testing, we can say that it is a science for evaluating the planting value of the seeds. What is the planting value? Planting value is nothing but it is only the economic value of seed lots to raise a good commercial crop. Until and unless a good crop cannot be raised from the seeds bought from the seed lots, concerned seed lots, their economic value will be defeated. So it is determined, the PLS value, it is determined, planting value of the seed lot is determined by the PLS value, that is the pure live seed percentage. And PLS, pure live seed percentage, is calculated by multiplication of the pure seed percentage to that of the germination percentage. So if we have a pure seeds, more number of pure seeds, and the germination percentage is up to mark, I mean to say that that depends on the different class of seeds. For example, in case of breeder seeds, it must have 80% germination, otherwise it will not be prescribed, not be good. So it determines, the seed testing determines actually the genuineness of a cultivar. It means the cultivar, the seeds must be true to the types of the particular varieties. So genuineness of the cultivars in respect of their physical, structural, physiological, genetical and biochemical properties. These all things covered under the seed testing programs. So if we talk about the objective of seed testing, it first determines the suitability of seeds for their planting value. Second one, to identify the seed quality problems. We have some problems. If we have some problems in seed testing and different quality of seeds, during testing we can identify the problems, what are the problems and their probable cause, what is the probable cause which affects this particular seed quality characters. Third one, to prescribe the specific procedure for drying and processing. If we have a high moisture content in the seed samples, we must prescribe the drying procedures and the different processing procedures. So next one, to observe the established quality standards of seed and labeling. Especially we know very well there are certain standards for different quality seed quality components. So it's after doing the seed testing, we can only observe the established quality of the particular seed lots. And last one, that it provides the basis. It provides the basis for price after establishing the seed quality. If a seed is not up to mark, is not a good quality, the price would not be much more or much more, you can say the very less price can be attained. On the other hand, if a good quality seeds is provided to the farmers, we can get a high price. The importance of seed testing, it was realized very much earlier, too much earlier, 100 years ago. For the planting value, we are already discussing about this seed testing. The seed testing have been developed to and agriculture and avoid some of the hazards of crop production. Why? Because if seed has not proper germination percentage, then the farmers may not raise a good commercial crop due to germination loss. So these are some of the importance which can only be taken care of by the seed testing. So these are some important. These are some historical background. It must know that the historical background of seed testing. The first seed testing station was established in Germany in the year 1969 under the direction of Friedrich Nove, one of the very good scientists in the seed testing and seed testing research. In 1871, seed testing laboratory was established in Copenhagen in Denmark, and next in India, the first seed testing station was established in the year 1961. And for all those seed testings, procedures and guidelines, a handbook on seed testing was written by the Friedrich Nove in the year and published in the year 1876. There are some of the institutes which are very much associated or related to the seed testing procedures and guidelines. First and foremost is the ISTA, International Seed Testing Associations, which were established in the year 1924, and time to time they provide the guidelines for different testing of the seeds. And some of the other agencies like AUSA, Association of Official Seed Analysts, are there. SCST, Society of Commercial Seed Testing, Seed Technologists, and Commercial Seed Analysis Association. These are some of the associations, some of the agencies which are related to the seed testing procedures and guidelines. So in case of before seed testing, for the purpose of seed testing, a seed testing laboratory is very much important. So we can say that it is a basic unit or it is a hub for testing a different quality components of seeds. The seed testing laboratory must be established with full-fledged, full infrastructural facilities. There must be sufficient manpowers, the equipment, sufficient updated equipments, the recent equipments, and the evaluating the planting value of the seeds should be there. The seed testing laboratory, the first and foremost objective of the seed testing laboratory is that the evaluating the planting value of the seeds, and the last one is the reporting results in time. It's very much important because when the completion of the seed testing is completed, then the results must be communicated to the concerned agencies within a limited time frame. Otherwise, it will be defeated. The purpose will be defeated. According to Kahra et al. In 1975, they enlisted five points of a good seed testing laboratory. First, that the highly responsible person should continue the work in absence of the staffs. We know that if any technical staffs are on leave, so in that case that the work should not be suffered. In that case, the highly responsible person must be substituted to continue the work. Next, the uniformity in equipments, procedures and results interpretations. There are number of seed testing laboratory working for the seed testing purpose. So in all those seed testing laboratory, the equipment should be uniform. Their procedures of testing must be uniform, and also the interpretation of results must be same. The seed testing laboratory should follow these points. Next point is the cordial environment among staff. It's very much clear to all of us that everywhere there must be cordial environment among the students or even among the staffs to continue a work in very, very effectively and efficiently. Explanatory remarks in reports by the leaders. The leaders should report the results with explanations, in a proper explanations. They also mention the procedures which have been adopted in the particular test. Quoting practical question for the research and further improvements. It's very, very much essential that if we want to have a good program, the research and further improvement in the seed testing laboratory, a practical question must be quoted in the results. What are those activities done during the seed testing? management of seed testing work. So we know very well that there are different types of seed samples. We know that the submitted samples is those samples which are submitted to the seed testing laboratory. So the submitted samples is received in the seed testing laboratory, and that seed samples is registered with all the information which have been mentioned in the particular seed samples, and also what are those required test is being done. It's also registered properly in a register for further process. Second, the moisture test. First, this is very much important because the seed samples received for the moisture test must be properly sealed. Otherwise, the proper result cannot be expected. Next one is the working sample, preparing working samples. Working samples is very much important. What are those? This is the reduced representative of submitted samples. We know very well that working samples are prepared in seed testing laboratory from the submitted samples, and that are the reduced representative of submitted samples. So for all those tests which have been mentioned by the concerned agencies, working samples has to be prepared, and for all those tests a card is attached for this purpose. Next one is the routine test. There are some of the tests which are done in the seed testing laboratory on routine basis. For example, in case of the seed purity is done on routine basis in the seed testing laboratory. Seed purity, particularly the physical purity test. The moisture test is also done on routine basis. If the seed samples are submitted for the issuance of certificate of certified seeds, then in that case the presence or absence of disease infestations is also considered as the routine test. Next, the reporting results is very much important, that after completion of seed testing or concerned seed test, the results must be communicated to the concerned agencies within 21 days of times. Otherwise, it may be, in any case, it should not be more than that of the 30 days. So, the reporting results is very much important, that if we provide the results in a short span of time, it's good for the concerned agencies. The storage of guard samples. What is guard samples? Guard samples must be understood that in the way that if any queries or any claim is done by the concerned agency with respect to the particular test results, then in that case the samples should be stored as a safeguard, and that is also considered as the guard samples, so that we can again retest. The concerned agency has to pay for the retesting, and then the retesting of the same samples would be done, but that subjected to the samples must be stored properly. Otherwise, if it is not stored properly, the same result cannot be expected. Last one is the maintenance of records. The records of results which have been given to the concerned agency must be maintained properly, so that after one term, within 1 year or after some period of time, we can just again provide the results to the concerned agencies or some farmers. Seed purity analysis. And during seed testing, it is the first and foremost test of seed purity. is the estimating the extent of biotic and abiotic impurities present in the seed lot of a variety. We know very well the seed lot. Seed lot is the physical lot, physical appearance of a particular seeds stored in either in bulk or in a bag. We know very well that the presence of impurities in the particular seed lot is estimated in the seed purity analysis. The testing of working sample is done as per the ISTA rules. We know very well that as per the Seed Act, a variety can be defined as a subdivision of kind identified by growth, yield, plant, fruit, seed, etc. As per the International Code of Nomenclature, variety is also defined as an assemblage of cultivated plants which is clearly distinguished by any characters. It may be due to morphological, which may be physiological, chemical or cytological, and when produced sexually or asexually and retain its distinguished characters. So we know that different characters, distinctness, uniformity and stability we know very well. So the variety must be novel, the variety must be uniform, the variety must be distinct for a particular characters to one to another. The objective of seed purity testing is first to determine the physical impurities present in the seed lot, the percentage of their impurities or dockage, and second one is to know the genuineness of a genetical purity percentage of a particular variety. It means that the trueness to the type of the variety in a seed lot. So physical purity testing is the test of the seed lots for their determination of different components. That is the pure seeds, the wheat seeds, the other crop seeds and inert seeds. And here we must understand properly the pure seeds, to whom we should call the pure seeds. Pure seeds refers to the species as stated by the senders. For example, if the senders give me the seed of maize, so we concern only the Gia maize. The maize, all the species Gia maize, we concerned with this. And if that may be the white maize or the yellow maize, we don't go for this purpose. So we can consider it as a pure seeds. It includes all varieties of that species. We say that if there is mixture of variety, we concern these differences not considered unless it is transformed to the fungal sclerotia, smut balls and nematodes and galls. If these conditions are subjected for the pure seeds. However, if removal of entire seed coat in leguminous crop, it is not the pure seeds. If a caryopsis size is 1/3 of the length of the palea, regarded as the pure seed in case of Gramineae family. The second component of physical purity is the inert matters. Inert matters are those matters which can be apparently appear. Apparently includes readily apparent, but you can see by our naked eye, and all those structure not defined as the pure seeds is considered as the inert matters. So during threshing we can have some stone particles, we can have some sand particles in the seed lots, that is considered as the in the inert matters. Florets are smaller than that of the minimum prescribed seeds should also consider under the inert matters. Sterile florets attached with the fertile florets is also considered in the inert matter components of the physical purity. Undersizes that the less than half of the original seed size is also considered in the inert matters, and if entire seed coat is removed in case of Leguminaceae and Cruciferous family, that is considered under the inert matter component of physical purities. Other crop seeds, that if in a seed lots there are the mixture of different the seeds of different crops. That is considered under the other crop seeds. Weed seeds. Weed seeds are those seeds which are legally approved as a weed seeds. For example, we can have an example that earlier the lantana camera was introduced from other part of the world as a ornamental plants, but later on it is designated, it is legally approved as a weed seeds. So it is the weed seed that must be legally. The procedures of physical purity test. There are the design procedures for physical purity test, and some of the apparatus required for this is the purity workboard, the forceps spatula, hand lens of different magnificence, stereoscope, microscope, balance, seed pens and sieves. The procedures are those: first we prepare the working samples. We know that the preparation of working samples, that the reduction of seed submitted samples by the different mechanical means the use of gamete divider or use of Boyer divider. So placing the samples on purity motherboard in a piles. The seed samples, the working seed samples is placed on the working motherboard in a piles. It means that it is arranged in such a manner that each and every seeds, the components of each and every seeds can be clearly observed, and then they are separated. They are separated by with the help of a spatula and our hand forceps. And the different physical purity components are just separated, and then they are weighed, and then their duplicate test may be done done to check the similarities. Weighing of all the components in gram with prescribed decimal places is done. So this of physical purity test, and this is the physical formula of physical purity test, that the pure seeds divided by the total seeds that constitute the pure seeds plus other crop seeds plus inert matter into hundreds. The physical purity test required for the seed certification. This is the standard of physical purity test, that it must be 96 to 98% in case of foundation and certified seeds. The physical purity test and the seed testing. In next class we will discuss about the genetic purity test. Thank you very much.